All right, before we begin the next section, let's start with a little review. For which of the clinical samples would it be necessary to perform an initial culture using a selective plate or culture medium to look for the presence of a pathogen? Would it be A, blood, B, cerebrospinal fluid, C, a stool sample, or D, synovial fluid? Go ahead and pause the video, think about that one. Okay, in this case, it would be the stool sample, right? Because that is coming from a non-sterile source. All of these are from sterile areas in the body, so you don't need to use that initial selective plating. Okay, so we talked about collecting samples. Let's talk about microbial identification using different tools, particularly biochemical, and we'll even talk about protein profiling. So we generally identify things using a series of tests, and we call this an algorithm because we follow a set path through this. We'll talk about broadly about some differences in doing this in gram negatives and gram positives. Again, we'll hit selective and differential media. We've had those several times before, so hopefully it's review. And then we will talk about a newer technology where we can actually look at all of the proteins in a microbial isolate and use that as like a fingerprint to rapidly identify what the pathogen is. Okay, so as we've talked about, labs have to quickly identify pathogens, right? Speed can save lives. If we know what is infecting someone, we can treat it much better. So how do they do this? Well, the spoiler is, right, you have to know about metabolism, biochemistry, boring things for sure for some of us, but very important properties. So let's look at this case study. We have Cindy, she's 38, and no previous medical issues. Goes into the ER complaining of a sore throat that's lasted about 3 days. She has joint pain, myalgia, muscle pain, and a fever. The day before, she had had a headache, neck stiffness, nausea, vomiting. Can you see where this is going? We've seen that before. She's not taking any medications, doesn't have any allergies, doesn't smoke, and lives with a husband, 2 children. All of them are perfectly healthy right now. So I have no pictures, no clues for you here. This is a black box, right? This is kind of the reality of what's going on. I think we have seen some of these things before, so hopefully you're starting to suspect meningitis, right? Cerebrospinal fluid is collected. That stiff neck, common sign of meningitis here. So we do a spinal tap there. Look at the CSF. It's cloudy when it should be clear. So that's like directly looked at. So we can look at that instantly. We can pretty rapidly do the white blood cell count and some other tests in here. We can also quickly do a smear and identify the bacteria on there. They do the gram stain. They're gram-negative rods. We then also grow the microbe, and we don't grow it on one medium, we grow it on many. So we grow it on chocolate agar, blood agar, and hectoan agar for identification. All of this points towards bacterial infection, and this is the slowest part, right? The growing of it. For this case, we use what we call a problem-solving algorithm to a series of step-by-step problem-solving methods. A lot of these are like yes-no tests. So remember the dichotomous key where if it's this, you go this way. If it's that, you go that way. It's kind of working off that. So for this case, we ask questions. Is an organism seen in the CSF of a patient with symptoms of meningitis? Yes, we did see it there. Is the organism gram-positive or gram-negative? We found gram-negative. Does it acid-fast stain? That's for mycobacterium. Remember tuberculosis, things like that. We found gram-negatives. So we have a pool of common culprits of meningitis. Neisseria meningitidis, Streptococcus pneumoniae, Haemophilus influenza, and Escherichia coli. So a lot of tests are being done all at the same time to give us information. We have the gram stain, we have plating on these 3 different types of plates. So we've limited the number of microbes. We have 4 possibilities. Let's use our algorithm to work through which one it could be. Okay, so our culture-based testing. We've plated it on differential and selective media. Chocolate agar has lysed blood cells, so there's no real chocolate in there. So this supports what we call fastidious bacteria, things that need lots of special nutrients. We know from previous information that all 4 of our pathogens should grow well on chocolate agar. But we also plated them on a different type of agar called blood agar, which contains whole blood cells, not lysed blood cells. We know that Neisseria meningitis and Haemophilus influenza will not grow well on blood agar. So we'll take that information in a moment. Hectoan agar, we have lactose, bile salts, things like this, so selective and differential. Only E. coli will grow on the hectoan agar. So in this case, our information, we have gram-negative rods. That rules out Neisseria. It's a diplococcus, and S. pneumoniae is a gram-positive. So that's our first part of our algorithm, right? Which of these are positive? Which of these are negative? What are their shapes? Then we look at the plates. It grew on blood agar. That eliminates Haemophilus influenza. And it grew on hectoan agar. That means it's most likely E. coli. So based on all of these different tests, we've narrowed it down to some like yes-no questions that point towards E. coli. If we needed to, we could do additional testing to further confirm this. And so that's like a very simple set of tests, right? You might not think so, but that's like limited. There are larger test suites that we call probabilistic indicators and analytical profile indexes. Here's an example of an analytical profile index that tests 20 different metabolic processes. So you grow up the culture and you add it to each of these little wells in here, and some sort of reaction will occur. Here you can see a positive versus a negative in this one. And so using these differences here, we can take this information and put it into a computer and get a probability of what the microbe might be. So this is a more comprehensive way. This takes overnight incubation to do it, and it uses all those biochemical things. You can further improve on this by automating the process. So using some sort of robot and computer to test and read these things, or replica plater type things to do this. So machines can handle more samples, more tests, and they make less mistakes. That's key. But it's still working on the same principles of this yes-no kind of thing. We could put this into a dichotomous key, right? We talked about these for certain traits, but this is for things like, can it use glucose? Negative, it might be Pseudomonas, or positive, right? Can it use lactose? Yes or no? Can it make hydrogen sulfide? Yes or no? Blah, blah, blah. Testing all these different things. And we can work through this yes-no set of questions all the way down to different types of bacteria. Yersinia pestis is here, E. coli is there, Klebsiella there, so on and so forth. Most of those tests I talked about are for our most common pathogens, which tend to be gram-negative, what we call enteric bacteria. The non-enteric bacteria require more complicated tests. There might be like special growth factors that need to be added to test them, but the principles still hold. So it just gets more complicated, but we start generally with the most common things, which tend to be gram-negative enteric bacteria. Gram-positives, particularly what we call pyogenic cocci, things that cause pus-forming infections and are coccus-shaped. We can look at other tests. Catalase test, does it have certain enzymes that can cause a reaction? We can put these on a microscope slide. Coagulase, another enzyme in there. And also that process we talked about previously called hemolysis. It's actually a couple different types of hemolysis. Alpha hemolysis versus beta hemolysis. Alpha hemolysis is actually due to hydrogen peroxide that is oxidizing the heme ions in the blood, whereas the beta form is when that toxin was made and it punches holes in the cell. So the red blood cells are actually being destroyed in beta hemolysis. Again, these are all algorithmic tests here. We even have some specific ones just for like certain really weird bacteria, right? Remember the mycobacteria, they don't really stain gram-negative, gram-positive, and they grow really slow. So we have acid-fast stains for them to identify things like Mycobacterium tuberculosis or leprosy. And there's special mediums. I think this one looks really cool. It looks like slime or something. It's this Lowenstein-Jensen medium for selectively growing these slow-growing mycobacteria. All right, I want to talk to you about something that is so cool, science fiction-y, if you will. This is called a mass spectrometer, and you've probably heard of these because every single TV show, the lab person always has a mass spectrometer. And a mass spectrometer can identify different chemical compounds based on the various principles that it uses. There is one type of mass spectrometry called matrix-assisted laser desorption ionization time of flight mass spectrometry, or MALDI-TOF MS. What this does is it can look at the protein composition of something that we put into it. So we have a clinical isolate of bacteria and we put it into the machine, and the MALDI-TOF identifies all of the proteins that are in there. And it can do this in an afternoon. I can then compare that to a database. So we put in our bacteria, it runs through this mass spectrometer, and we get out this reading that's like a fingerprint for different bacteria. So in this case, this matches up with E. coli. So this is going to change the way we identify microbes. This is still rather expensive and specialized technology, but it will become much, much more common in the future. So we still use plates, right? We still can start from a plate, but we have new technologies that work as well to identify microbes. All right, so remember we talked about selective media that inhibits the growth of one group, permits the growth of another group, whereas differentials, they show some sort of difference distinguishing between two types of microbes. Identification through biochemical tests, we talked about dichotomous keys and automated systems, and we talked about hemolysis, right? That's another trick we can use, the acid-fast stain, and that really cool technology called mass spectrometry. That's it for 25.2.